Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton today, guys, and we are going to be talking about more A21 biology, guys. We're getting towards about, I think, maybe 3/4 of the way stage, I suppose you can maybe talk about. But yeah, last time out we covered the eye, just the basic stuff. But today we're going to talk about the nitty gritty, the retina, the rods, and the cones. More so the rods and the cones will be in very much detail in our next video where we're going to specify about convergence and all that there. But today we're really just going to talk about how the retina can be used for its various functions and how rods and cones are quite prevalent on that. So we're going to get straight into this. So the retina has two layers. The first layer being photoreceptors, which are the rod and cone cells, and this is where light is actually detected. The retina is a complex structure which contains different types of nerve cells, and a large number of fibers form a sensory neuron that collect to form the optic nerve. Rods and cones will synapse with the bipolar cells, which connect to ganglion cells by a synapse. The axon from the ganglion cell will collect to form the optic nerve, and this will take information to the brain. Behind the retina is the layer called the choroid, and this layer is pigmented with cells that contain black melanin, which absorb light rays, preventing the light being reflected back through the rods and cones, which will cause a fully distorted vision. The retina is inverted, which allows light to pass through the bipolar and the ganglion cells before reaching the photoreceptors, and this inversion explains the occurrence of the blind spot, with fibers forming the optic nerve starting inside the retina and that crosses the surface, forming the optic nerve passing through the retina where there's no room for rods or cones, and therefore there's no photoreceptors and it's not sensitive to light at the blind spot. So vision and dim light then. Well, rod cells are what allow you to see in low light, okay? And they contain membranes which are stacked parallel. They have this pigment that's called rhodopsin, and it's composed of two compounds known as opsin and retinal, which is from vitamin A. When light strikes rhodopsin, it will break down into these products called opsin and retinal, which give the generator potential. And this breakdown will cause the rod to become less permeable to sodium ions, and therefore less sodium ions will diffuse in and more will be pumped out, meaning the inside will become more negative. And this hyperpolarization is the generator potential. So the bipolar cell will then release the transmitter into the synapse, and when sufficient is released, an action potential will be generated. This is all called dark adaptation. So essentially what you need to know is that rods are composed of rhodopsin, which is composed of opsin and retinal, and whenever light strikes it, it has a generated potential created, which means that it is less permeable to sodium ions, as less diffuse and more diffuse out, creating a generator potential. So rods are sensitive to light as rhodopsin absorbs light readily and easily broken down, and the consequence of this rods also display retinal convergence, and the consequence of this is that the brain does not distinguish which rod of a group shares the same optic nerve which has been stimulated, meaning there's decreased visual acuity as there is a lack of ability to discriminate detail and color. What this technically means is that there's like 4 different rod cells for one ganglion cell, for an example, or for one ganglion thing, which then leads to the optic nerve. So essentially, there's 4 rods that share the same optic nerve, is essentially what I'm saying, and therefore the brain cannot tell which one is being stimulated, and therefore it can't have high visual acuity, or can't talk about color or detail. Rhodopsin can be resynthesized by having ATP, which is supplied by the mitochondria, and after 30 minutes in complete darkness, all the rhodopsin will be reformed. So vision and bright light. Instead, you have these things called cones, and these have a pigment called iodopsin, which is less readily broken down than rhodopsin, and these only produce a generated potential and bright light. Cones do not exhibit convergence, as there's one cone for one neuron, with each cone generating an impulse to the brain, which means there's high visual acuity as the brain distinguishes between points close together. Iodopsin is free-formed, which are each sensitive to different wavelengths corresponding to different colors such as blue, green, and red. This forms the basic trichromatic theory of color vision, where pure red light only breaks down red iodopsin and only red cones fire impulses. Thanks for watching this video by C.A.R. Entertainment. I've been Dylan Hamilton, and I will see you next time.